Hi everyone, and welcome back to STEM Revise. In today's video, we're exploring three essential polysaccharides: starch, glycogen, and cellulose. Each of these molecules plays a unique role in living organisms, from energy storage in plants and animals to structural support in cell walls. We'll break down their structures, functions, and the differences between them. Let's jump straight in. So let's jump into starch, the primary energy storage polysaccharide found in plants. Starch is vital because it allows plants to store energy efficiently for later use. Starch is made up of long chains of alpha glucose units, linked by glycosidic bonds. There are two forms of starch, amylose and a myelopectin. Amylose is unbranched, consisting of alpha 1,4 glycosidic bonds, and it forms a helical compact structure. This compactness makes amylose very efficient for storage, as it takes up minimal space. A myelopectin, on the other hand, is branched, with alpha 1,6 glycosidic bonds at the branching points. This branching gives the myelopectin multiple endpoints, which enzymes can access quickly in order to break down the starch, making energy release rapid when needed. The compact helical structure of amylose, along with the branched form of a myelopectin, makes starch a highly efficient storage molecule. Another crucial feature is that starch is insoluble in water. This property is important because it prevents starch from affecting the water potential within cells, so it doesn't cause water to move into or out of cells by osmosis. Additionally, since it's insoluble, starch won't diffuse out of the cells where it's stored, ensuring energy is kept available in one place. When a plant needs energy, starch is hydrolyzed, breaking down into its individual alpha-glucose units. These glucose molecules are then easily transported and used in respiration, where they provide the energy necessary for various cellular activities. In summary, starch's structure with both amylose and the myelopectin, its compact, insoluble nature, and its ability to release glucose quickly makes it an ideal energy storage molecule for plants. Now, let's move on to glycogen, the energy storage polysaccharide found in animals and some bacteria. Glycogen is very similar to starches and myelopectin, but it has a key difference that makes it suited for its role in animals. Glycogen has a structure that's much more highly branched than a myelopectin. While a myelopectin has branches formed by alpha 1,6 glycosidic bonds, glycogen's branching occurs more frequently, making it even more compact and allowing for a larger amount of glucose to be stored in a small space. This highly branched structure is what distinguishes glycogen from plant starch. Glycogen is primarily found in animals and some bacteria, and is especially concentrated in the liver and muscles. These organs use glycogen as a readily available energy reserve. Unlike starch, which is used for storage in plants, glycogen is specifically adapted for the needs of animals. Like starch, glycogen is insoluble in water. This is important because it prevents the molecules from affecting the water potential in cells, ensuring there are no unwanted osmotic changes. Its compact structure also means that a large amount of glucose can be stored efficiently without taking up too much space, making it ideal for animals that need quick energy storage. The highly branched nature of glycogen is particularly beneficial because it allows for rapid enzymatic breakdown. The multiple ends where enzymes can act make glycogen easy to break down into glucose when energy is needed quickly. This is crucial for animals, especially those with high metabolic rates, like mammals, as they need a fast and efficient way to release glucose for energy, particularly during exercise or stressful situations. So to summarize, glycogen's highly branched structure allows it to store a lot of glucose in a compact form and break it down rapidly, making it the ideal storage molecule for animals with high energy demands. Finally, let's take a look at cellulose, a polysaccharide that plays a crucial role in plant cell walls. Cellulose is made from beta-glucose monomers, which form long, straight, and unbranched chains. The key features of cellulose is that the glucose molecules are linked by beta-1,4 glycosidic bonds, which causes the chains to be straight rather than helical. These chains run parallel to each other and are held together by strong hydrogen bonds, which create a network of crosslinks between the chains. These crosslinks form microfibrils, which are very strong and contribute to the structural integrity of the plant cell wall. Cellulose is a major component of plant cell walls, and its primary function is to provide rigidity and structural support to the plant. Without cellulose, plants would not be able to maintain their shape or withstand turgor pressure, which is the pressure of a cell's content pushing against the cell wall. The microfibrils group together to form larger fibers, which further strengthen the cell wall. The arrangement of microfibrils in the wall gives it incredible tensile strength, which is essential for plants to stand upright and resist physical stress. The strength of cellulose comes from the extensive hydrogen bonding between the beta-glucose chains. This creates a tough, rigid structure, but the organization of the microfibrils also allows the cell wall to be flexible. This balance between strength and flexibility is vital for plants, as it allows them to maintain their shape while also growing and expanding as they develop. So to summarize, cellulose's straight, unbranched chains of beta-glucose form strong microfibrils through hydrogen bonding. This contributes to the plant's cell wall's strength, flexibility, and rigidity, providing essential support for the plant.